In this research, I'm looking to investigate whether microRNAs can be of therapeutic benefit to individuals with CDKL5 deficiency disorder, which is also called CDD. And CDD is a rare neurodevelopmental disease, which is caused by mutations in the CDKL5 gene. And this gene is highly expressed in the brain and is involved in processes such as proliferation, the formation and growth of neurons, and it is very important in the development and function of the brain. So in cases such as CDD, where there are mutations in this CDKL5 gene, this reduces the amount of the functional CDKL5 protein or alter its activity in neurons. So this disrupts the development of the brain, and this results in the features which are seen in CDD, such as early onset epilepsy, intellectual and motor disabilities, and cortical visual impairments. And so currently there are no effective treatments. The only treatments available improve the symptoms rather than addressing the underlying cause of the disorder. And so for the last few years, the Future in Our Lab, where I'm currently doing my PhD, has been working in epilepsy research, and they have found that there are these key brain functions are controlled by molecules called microRNAs. And they have been able to show that these microRNAs are able to improve motor control and either stop seizures or prevent epilepsy in animal models. So my focus in this project will be on microRNAs and to test whether they are involved in the regulation of CDKL5 expression. I will be using a novel CDKL5 knockout mice model. And firstly, I want to establish that this model actually has CDD. So I will define its motor and cognitive deficits, which are seen in the patients. And this will be done through a comprehensive battery of tests for natural behaviors, anxiety, motor control, and learning and memory. And then I will hopefully be able to identify any dysregulated microRNAs in these models. And then when I will do that, I will design microRNA inhibitors and deliver them to these models. And what I want to do is determine that the effects of these microRNA inhibitors on the behaviors of mice. And this is to check whether they can prevent the features of CDD. I mentioned that there is no treatment for CDD. It is really a devastating disorder with children having severe intellectual disability, and most of them can't walk or talk or eat by themselves, and many are confined to a wheelchair, and there's very little that is actually known about CDD right now. So with Epilepsy Ireland as my enterprise partner, I'm hopefully trying to firstly build a comprehensive understanding of the disorder and get some positive results. And when I get those, I will report back on those and hopefully publish them. And all being well, this will eventually drive the development of advanced new precision therapies. And also, as the first PhD student in Ireland who's working on CDD, I'm really looking forward to working with Epilepsy Ireland to raise awareness about it and get patient and public involvement in my research, because this will give me more important insights into how I can make it more relevant to the needs of patients and the carers. And this is extremely important to me as a researcher, because I want to make sure that my research results in a real change in the world.